Hi, my name is Maya Rosenfeld, and today I'll be explaining a DNA replication method called PCR, which is short for the polymerase chain reaction. Now, people say that science is a bit like cooking, and I didn't really understand that until I found out this sort of DNA replication method, and I think this is one of the best ways to explain how that works out. So this process involves a short sequence of double-stranded DNA, primers, heat-resistant enzymes, of which the most commonly used is known as Taq polymerase, and DNTP, which are all the nitrogenous bases of DNA. All of the contents are put into a series of small tubes that go into a PCR machine. This machine performs the thermal cycling needed to complete each PCR cycle. The first step of PCR we call denaturing. This step involves heating the contents to about 96 degrees Celsius, which separates the double-stranded DNA into two single-stranded helixes. The second step of PCR is called annealing. The contents are cooled down to about 55 degrees Celsius, after which the primers, which are short complementary sequences to the target sequence, bind to the 5' end of one helix and the 3' end of the other helix of the target sequence. The third and last step of PCR is called extension. This is where the contents are heated up to about 72 degrees Celsius, so the Taq polymerase is able to extend the primer and build the rest of the complementary sequence to each of the single helixes with DNTP, which is also known as the building blocks of DNA. The building blocks of DNA consist of the known nucleotide bases G, C, A, and T. A single cycle produces 4 double helixes of the target sequence. This cycle is then repeated at least 30 times to have a sufficient amount of cloned DNA. So, as you can see, PCR is a really small cycle, but it plays a really major role in things such as recombinant DNA, which is combining DNA from different organisms and putting them together. And so I hope this video shows just how big a role such a small cycle can play.